Hi class, in this lecture we are going to be looking at replication. Replication is making a copy of DNA to split itself into two and build two new strands. Alright, so again, what we like to think of is that replication occurs in both strands simultaneously, and this creates complementary strands. What I want you to think about is that next time when you unzip your coat, this is like DNA unzipping. The zipper itself is the enzyme helicase. Helicase will unzip the DNA and open it up. Once that is open, now you have the room to build two new strands. We call that semi-conservative. So the idea is, if you look at this diagram over here, you can see the blue strand represents the old strand. The purple represents the new strand. This is how it's built. This is a semi-conservative process. What this means is that you always have one old strand and one new strand. This is because if you make a mistake, you can always go back and fix the mistakes using the old strand. So that is the reason why we always like the semi-conservative, because it opens up, you build two new strands, and you have a way to go back and fix it. Alright, so in DNA replication, we are making an exact duplicate of DNA. This process involves about 30 enzymes. I do not want you to know all 30 enzymes. We are going to talk about a few of them here, which are involved in opening and making copies of the DNA. The one that I want you to know is helicase, because helicase is essential. It unwinds and unzips the DNA double helix. Remember, this is all about making a copy of DNA. In bacteria, this begins at the orange of replication. This is like the site where the zipper would start and stop. So that's the orange of replication. The enzyme will open up the DNA at that site. Remember, the first thing that happens is that helicase will unwind the DNA. So what happens is helicase binds to the DNA, breaks the hydrogen bonds, works its way around. Then there's another one going on the other side, and eventually they will meet in the middle, and now you have two new strands. At the start, the range of replication, an RNA primer is synthesized by primase. This primer is added to the DNA, and so now you can build the new DNA strand. The primer is represented in green. The DNA polymerase binds on and builds the strands. So you can see these shorter little segments, which is called the lagging strand. This is because of the orientation of 5' and 3'. DNA can only be built in 5', 3' manner, so the opposite side has to be built in the opposite direction. This is one of the challenging things about DNA. Since this is microbiology class, so I do not like to get bogged down. However, I want you to understand the process. I talked about that DNA polymerase III adds nucleotides in 5' to 3' directions. So the leading strand is the strand that gets built in one direction. It is synthesized continuously in 5' to 3' direction. The lagging strand is synthesized 5' to 3' in short segments. So the leading strand gets let out from the orange, whereas the lagging strand is coming up from behind. Then you have other things. DNA polymerase I removes the RNA primers and replaces them with DNA. When replication fork meet, the separation of the daughter molecules is complete. Next, ligases links the DNA fragments along the lagging strand. Then eventually, it makes one continuous piece of DNA. So this is how the DNA polymerase I and the ligases will remove these primers and fill this in and build a new DNA, so that the lagging strand get all into one piece of DNA. This shows you that the helicase opens it up, then the lagging strand makes smaller little pieces, and the leading strand gets built in the correct orientation. I have a video to show this here. If you are not getting it quite clear, watch the video and it will make a lot more sense.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
This is an excellent little video, and I talked about this. The lagging strand has to be built in the little segments, and we call these segments Okazaki fragments. Again, helicase is responsible for unzipping. The strands are built from 5' to 3', and 2 new strands are made from the 2 old strands. We call that semi-conservative replication.